told me about this, I don't think my feet touched the ground for most of the rest of the day. And then a funny thing happened later in the day, and I said, what the hell am I going to talk about for an education award? Especially when I became aware. Pardon me? Put it higher. Especially when I became aware of who else was receiving awards today. And I consider myself very much a blue-collar kind of worker. I really fretted about this for many months. I finally realized that I really had never developed any new methods, and on the few occasions when I thought I discovered something new and then I went to the literature, I realized almost everything had already been published, and whether or not it was not brought into popular literature because it was done on an amphibian as opposed to what some people would have thought was a real animal, I don't personally believe that, like a chicken, which I later found out most people in medical school still don't believe is a real animal. Or whether it was the language that our ancestors, predecessors used, that maybe just became old-fashioned and all the good work went out of vogue, whatever. So I decided I would draw upon the fact that since I worked with Viktor for several years, and he was an invaluable mentor to me, not just because of the science, but because of the philosophy. And how he viewed science, and the fact that how he viewed science was based on how his two favored predecessors, Spemann and Harrison, viewed science, and how they learned, one of them from Brooks and Morgan, one of them from Bovary. And so I thought I would talk about this legacy of which I am a part, certainly not a creator, just a person who followed along in those footsteps. All right, so one of these buttons. So I chose the term legacy of experimental embryology. A legacy is something handed down or by or received from an ancestor or predecessor. It's a very Spemann-esque definition. It really says it's putting the focus on the signal rather than the responder, but nonetheless, that is the definition. That's what I'm going to talk about. And that's Viktor, of course, at this meeting in 1968 with Johann, his best friend and fellow graduate student in the early 20s, Johann Ho. And I have to also say that while they were my intellectual mentors, my passionate mentor was the chicken embryo. I told people that at a very early age I adopted a mistress, and not in any threat to the propriety of living, I kept that mistress, and even though I don't have any immediate contact with that mistress anymore, the memories are still very much with me. Please don't take that out of context. So in thinking about where should I begin, I took one of the things that used to characterize some aspects of graduate education is we were told to take courses, or I was told to take courses in the philosophy and history of science. And one of those courses was taught my senior year as a graduate student by Viktor Hamburger. One night a week we would meet in his home and he would tell us about the heritage of experimental embryology. You will see a number of quotes. I recorded those sessions, and I had not listened to those tapes since 1972. And I have most of what I'm going to say is actually stolen from Viktor Hamburger. Where to begin is in terms of how did it relate to what I actually ended up doing. I'm not going to go back to Aristotle as Viktor would. He spent a whole hour on Aristotle. But I'm going to talk with about Goethe. Goethe was an amazingly multidisciplinary, creative individual. He was a writer of popular stories. He was what we would call a journalist. He was a friend of dukes and emperors. He was a city manager who figured out how to fix the roads and fix the sewers. He was an avid reader also of philosophy. And he developed this concept called nature philosophy, or philosophy of nature. And what it essentially said was that every organism in some way strives for and reflects perfection. Now, the strives for is an interesting verb because it's very teleological. You know, if you're striving for something, it means you know the goal, like running a marathon or something. And that's very teleological. The other word in that is perfection. And this gets kind of tricky because perfection, in probably to some people today, but certainly in previous centuries, the man was never going to achieve perfection. If you thought about the concept of perfection, you linked it to God, the only source of perfection. And that was the essence of nature philosophy, in that science and philosophy were one. That unification, part of the German Romanticism, lasted for a long time. It defined Spain in terms of how he thought, and we'll come back to that. So how did this have anything to do with me? Well, you may know the story. One day Goethe was walking supposedly in a cemetery, and he found a skull of a deer. This is not that deer. This is a deer found in Tompkins County, where I live, because some hunter did not choose to take that part of the body home with him. But when he looked at this skull, and his was much more rotted than this one, what he noticed, if I actually turn this the right direction, the laser goes on the screen instead of my wrist. What he found, he said, well, gee, that's the basi-occipital, and no, there's a basi-sphenoid, and there's a pre-sphenoid, and there's a gap between them. There are segments. Holy moly! It's just like the vertebrae. The head is segmented. It's more perfect to think of it that way. This gave rise later on to the dogma of head segmentation. Notice I don't say the theory of head segmentation. It became, in the early part of the 20th century, the dogma. This is how the head developed. In every part was a reiteration of the one in front of it, and the one in the back of it. It would have a motor nerve, it would have a sensory ganglion, it would have a pharyngeal pouch or something sort of similar to that, and it would have what we might call abaxial type muscles and branchial muscles, and boom, 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 boom. A beautiful model. There was one problem with it, and that is that organism never existed. But parts of it, you could find, if you looked at enough organisms, you could find each of the parts somewhere. This really didn't come to the fore for me until a fellow who became a good friend of mine, Steve Meyer, worked with Betty Hay, and then did tilt scanning electron microscopy of the mesoderm, the avian embryo. And what he saw were a series of swirls. You can't see them if you don't have the stereo vision equipment, and little grooves. And he called each one of these a semitomere. And within a few years, he and Anton Jacobson and other colleagues found these in all these different animals. Now, there's some debate about whether they're real or an artifact, and I'm not going to try and get into that. What we do know is that if they exist, they are of no consequence in higher vertebrates, because nothing you look at, whether it be neural crest cells or motor axons or anything else that we know of happening and we manipulate these, nothing else follows that pattern. Maybe they're a vestige of the ancestral condition. So how does this play forward? Well, that's old, it's dead, who cares about that? Look in a minute in almost every medical embryology textbook at the larynx. And what you will find is that they'll talk about the first arch, second arch, neural crest arrived. They get to the thyroid cartilage, and they put that, it was fourth arch neural crest. It's logical. It kind of is close by other things like basihyoid and whatnot. And then you get to this one, the sixth arch. What's the sixth arch? There hasn't been a sixth arch since amphibians. But there it is. It's in the textbook. Why it is in the textbook still is not clear. 25 years ago, in the avian embryo, we mapped the cricoid neuranoid as mesoderm derived. Oh, it's that chicken stuff. Let's talk about real animals. Finally, when the Wnt1 constructs became available and they could label neural crest, they found it was found that the thyroid and the epiglottis or neural crest cricoid neuranoid are not. And I had seen this, got some of these animals from my friend Miles Epstein. I didn't ever amend, he didn't publish it, but there's this recent paper by your president, one of the most beautiful papers you will ever want to see about integrating experimental embryology and molecular biology and electrophysiology and microscopy. Everything you want to know about the development of the larynx in one paper. Beautiful. So this is wrong, that whole six-arch concept, but it tells us that these old ideas and dogmas die very slowly, whether they're right or wrong. All right, back to embryology. None of those people paid much attention to what Huxley said way back in 1868, that bogus. I had an intersection. I'll tell you one personal story. I actually intersected with Thomas Huxley. No, I'm not that old. I am emeritus professor, retired, but not that long ago. In 1991, Gillian Morris invited me to give a lecture at Oxford. And I have to tell you, I was pretty full of myself. Getting over, taking the train up to Oxford. Well, I didn't take it from Ithaca to Oxford, but from London to Oxford. I think, I'm at Oxford. This is it. And when they took me into the room to give the seminar, it was more like a big library with soft, cushiony chairs and whatnot. And oh, this is nice. And they had a table and there's a lectern on it. And I looked at the lectern, and there was this little plaque on it. It said, "In this room, on this podium, Thomas Huxley gave the first public defense of Darwin's theory of natural selection." If you've ever played with a kid and you blow up a balloon and you let it go and it goes, that's exactly what happened to me at that moment. Embryology became a common part of science, in large part because of two things. Darwin, because natural selection and variation were his proposal, but that has to occur before the adult. And that was a big black box. And Darwin knew this. There was nothing he could plug in there. Wilhelm Hess contributed to this, and I showed this picture because these were the two early describers of head neural crest cells. Wilhelm Hess didn't invent the microtome, he perfected the microtome. He perfected how to embed embryos. And he was the first one to section embryos and look at their innards. And that's from one of his early papers. The other thing he did was he invented camera lucida. And with this, you could draw, where you look through the microscope and you could draw what you saw, he would draw it, and then he would trace those onto wax plates and cut out the little holes where the lumen of the brain and whatnot, and he'd stack them all up, and he had a three-dimensional model of an embryo. And he befriended a father-son team named the Zieglers, and they knew how to make molds and fill them with wax and have models. And they worked together, and they made the first models of embryos. These models are of human embryos, and they were shown in 1884 at a Paris exhibition, you know what I mean, fair. And they were the first time the public ever saw a human embryo. And some people were outraged that someone would dare to show such a thing. Amazing guy. Francis Balfour, an incredible genius. Um, he took an embryo, when he was a sophomore in college, he took 2 courses. One was called Embryology and the other was called Evolution. And he put the 2 together and he said, this is what you have to do. You have to look at lots of different embryos of different species. He was a full professor at Cambridge by age 25. Oxford offered him a position and Cambridge offered him a department called morphology. By the time he was 30, he had published a 4-volume, 2,000-page book that had virtually every invertebrate phyla, all the classes of chordates, embryo drawing, 700 of his drawings. Unbelievable. Unfortunately, he died a year later. He and a friend went climbing Mount Blanc in Switzerland, which is very challenging, and they fell off and they died. Incredible amount of work that he did. The other, this is not an embryologist per se, but an anatomist, was Bateson. And Bateson took the stand that, okay, if selection requires variation, let's look at lots of variations. And he was most interested not in how species A and species B differed, but what were the variations within a species? And amongst the ones that he drew, very appropriate for the other speakers, was this blowfly. This blowfly has antennapedia. He didn't call it that. He just described what it was. He had another one with a honeybee. My favorite, nothing to just be disparaging towards arthropods. This is my favorite picture from that his 1894 book. This is from an 1851 paper. This is a young woman, and she has ears coming off of the 4th pharyngeal pouch. It is a homeotic transformation, probably. They did not actually clone the genes. They cut off the external ears, and she apparently had nothing else wrong with her. I mean, the internal anatomy was not wrong. The other neural crest cells associated with those arches were doing their normal thing, or she wouldn't have been alive. One of the joys that I had was being invited to go to the veterinary college. And what I was introduced there, when I first went to the vet school, they said, well, you know, we require you to teach. I'm happy, I like to teach. And said, oh, by the way, you're going to be teaching a 45-lecture course in mammalian embryology and congenital defects of domestic species, which was about 44 more lectures than I knew anything about mammalian embryos or malformations. But they were great mentors, and once I showed an interest amongst the pathologists, I ended up doing well over 1,000 postmortems on animals with defects. One day I got in the mail. Actually, my colleague, my predecessor, got in the mail and gave to me a box addressed to the embryologist at Cornell Veterinary College. And inside were 2 pickled Burmese neonatal kittens. And I looked at them, and this is a lateral view. It had no nose. Okay. And this is an on-face view where the nose was supposed to be, were these 2 things. Well, what the heck were those? And because it turns out that these animals were showing up at Burmese catteries around the country, we were able to get lots of them. We were able to breed them and get lots of embryos. This is a fetal stage. And what you can see, there's the maxillary prominence, there's the maxilla. These are supposed to be frontal nasal. They're maxillary. I was so excited to be able to publish a paper with the term homeotic in it, knowing nothing about any of these things other than mainly my conversations with Rob. We have no idea what it is. What we do know is when you look at very early stages, we did a scanning electron microscope, it has normal nasal placodes. It has normal looking neural crest cells that make a mistake and they think they're maxillary. Okay, back on track. August Weissman, I'm sure you know quite a bit about what he did, He was mainly a theoretician. He wanted to work with embryos. He had horrible vision. He couldn't see them. Couldn't do microscopy. But he came up with the germplasm theory. Extraordinarily influential. It was influential. Bovary studied with Weissman. And Spemann trained with Bovary, as you've already heard. It influenced one of his other students, was a young fellow who came to work with him, a rather strange fellow, independently wealthy, single-minded, essentially did whatever he wanted to because he didn't have to earn a living. And he spent a year with Weissman. He certainly knew about this. His family owned property in Naples, within walking distance of the Naples Marine Biology Station, which is the place you went to in the summertime to look at marine organisms in Europe. That fellow was Hans Driesch. And of course, Driesch's famous experiment separating the sea urchins challenged, to some ways, people's way of thinking, challenged Weissman's hypothesis about segregation of determinants. And when he first reported this, it was very straightforward. Regulation, they called it adaptation. It's a fundamental property of developmental systems. There has to be interactions among parts. This is brilliant. And we all believe this. There was a problem, of course, and that was that Driesch thought, well, okay, it's chemical. And what chemical process do I know that if you take away half of the chemicals, you end up with the same out? No. It's a pulley. It's mechanical, force and all that stuff, or a lever. Well, we've all been on seesaws. If you cut a seesaw in half, it doesn't have any of the same mechanical properties in fourth generation. And he went through every hypothesis, and then he came up with the fact that there is nothing that he knew about that could explain this, and it must be entelechy, an act of God, extra non-material. Driesch is the only one that I ever heard Viktor Hamburger say something negative about. And this is a quote. This is from the tape. It was a futile exercise based on the inability to distinguish forces not yet discovered from forces not yet discoverable, i.e., if he didn't know it existed, it couldn't ever exist. I mean, it sounds, but this was that whole philosophical basis, very prominent at the time. Spemann, a very complex individual. He was a student not directly of Goethe. He loved the nature philosophy concept. This notion of perfection, this teleological notion that the ends drives the means. Trained with Bovary, spent a lot of years with him, and I'm not going to go through his experiments. They're well known to most of you about, delayed nuclear acquisition and the fact that the position of fertilization affected the first cleavage in these species, and that then determined where the blastopore would form. And he knew that relationship shortly after the turn of the century. In the 1916, 17, 18, he published several papers that showed, because he was working on the blastopore, he showed that when you transplanted these blastopores, you got funny things happening in the belly, but he couldn't distinguish whether or not the transplant made the tissues that were the resulting tissues, or did it cause other things to happen, because they had no markers. When he went to Freiburg in 1919, took Otto Mangold with him and as an assistant, and he set up a lab. And for that first year when the lab was up and running, he had 3 graduate students: Hilde Prushold, Viktor Hamburger, and Johann Ho all came. She was the most senior of them because she had had actually more biological training than the others. And he gave her the project to work on regeneration in turbularia. It wasn't working out well. He tried to do them, and every time they turned this little beast inside outside, it would revert back to outside outside. And it was getting late in the breeding season for their salamanders, and he said, "Oh, here, try this." They had obtained amphibians with different intrinsic pigment contexts, so they could follow the transplant. Over the course of that season, the next year, did 250 transplants, 12 survived. Of those 12, half of them had something like this, the duplication. This, that's a picture from her journal. That is the organizer. And Viktor was in the room, second week in May 1921, when she brought in that specimen and showed it to the group. She was so excited, it lived. It took a while before the significance of everything really settled in. Okay. Just Stamen's explanation was linked to this natural philosophy, this teleological belief that the ends define the means, or the whole is always greater than the sum of its parts. Spamen believed very much. You could identify and you could experiment on each of the parts, but no matter how many parts you defined, you wouldn't really achieve the whole. Unfortunately, he didn't have the benefit of the mid-20th century or late 20th century sage, Larson, which actually gives a much more thorough explanation of exactly what the organizing center is all about. Johann Ho, very different than all of the other people that I will talk about. He came from what you would call a middle-class background. He wanted to be one of those natural scientists who went out in the field, collected organisms. He would draw their pictures. He was an artist. And he would come back, and maybe one of them had never been discovered, and he'd get to name it. That's what he wanted to do. So he had applied to go to Freiburg because the chairman of zoology in Freiburg was a natural historian, and this is what he had done. And he applied, but he asked for a year's deferment because he was doing other things and wanted to finish some studies. He showed up at Freiburg and discovered that the fellow who had been the chair had died and had been replaced by this guy named Spamen. And so Spamen realized that this was not a person who we wanted to get fancy experimental embryology. He gave him a project to look at the development of the liver in amphibians. Spamen was not really interested in the project, and the only one less interested in the project was Hofreiter. But Hofreiter was not philosophically inclined. When Mangold and Hilda and Viktor would go downtown for probably drinks, but they would go down and talk about Nietzsche and the meaning of life, he would be, Hofreiter went out with his artist friends. And very different. So they knew each other, but they were not chummy. He left very disenfranchised with science. He went out, spent 3 years getting odd jobs, doing a lot of portraiture. I think it's more like caricatures that he would draw. And Viktor at that time was working in Berlin with Mangold. Viktor was offered, Spamen wanted Viktor to come back as an assistant professor, and Viktor told Hofreiter there was an opening in Berlin, and he went back, he needed the job, and he did amazing things. The first thing he did was fix dorsal lips, put them into the blastocele, and he got a similar result to a live one, and various other things, which led Spamen to say a dead organizer is a contradiction in terms. He did fate mapping using Werner Voigt's newly developed vital dyes. And he would look at what, I saw these posters about collective migration. He didn't use the term, but he loved to look at cells, and he did exogastrally. They would come outside, and he would see how the spots would get bigger, but they would always stay cohesive. And then he did these dissociation reaggregation experiments where he separated cells of multiple neural stage tissues, allowed them to reaggregate, and they would sort themselves out. how does this have anything to do with what I ended up doing? He knew about the concept of cell-specific movements and adhesions. When I was doing mapping of, and I made lots of maps. Most of my career I made maps. And I mapped placodes. I mapped the placodes, and I mapped neurocrest, I mapped mesoderm. And these little diamond-shaped colors just indicate the leading edge of those tissues, early, middle, and late stages. And what you can see is they all start up dorsally, and they all end up laterally. And I don't know, at some point, I asked the staff to put out Oreo. How many of you got an Oreo cookie? They didn't put out my Oreo cookie. All right. If you pick up an Oreo cookie and you walk across the room, did the cream filling migrate? It went from point A to point B, but that cream filling didn't crawl. And I published this in 1984, and I have to tell you, not many people really liked the idea. I wasn't attacking the concept of migration. I was simply saying that in the head, most neural crest cells are just hanging on to one another, and they're moving through the dung of the neural crest cells that went ahead of them, and they're staying in registration with the cells around them. Okay. Hofrader's drawings. These are two of his drawings. When I visited Hofrader in Rochester, he was already 80 years old. And the wall beside his stairway going upstairs was lined with these etchings. I wasn't going to ask him what each one meant or anything, but these were some, and these were pictures that Victor had. Preformation, and the one on the right there, evolution, stop, don't go any further. I had no idea who Anarchy was. That was the Greek goddess of fate and destiny. And that's her sign posted saying, don't go any further. He was not a fan of stamens. These are drawings that he made. There's Spamen. That's an avian-looking Pegasus carrying an egg. These are Spamen's postdocs. I don't know which one goes with which. It's hard to see here, but you see his hand kind of over something. If you look very carefully, there's a two-headed chicken. There's a two-headed chicken. Those were the organizer experiments. Over here, he's got all his ducks in an order, except for this one. I assume that was his self-portrait, the duck facing the wrong way because he wasn't playing by the same rules, and Spaman's admonishing the one next to him to get that duck in line. I met Victor. He was teaching an anatomy embryology course. I TA'd. It was an 8-credit course. And he taught the anatomy part, not the embryology part. He came to the U.S. in the early 30s. He brought the Spaman tools with him and techniques to an avian lab and taught Lily's postdocs and Benjamin Way or others how to do microsurgery with tools that you would make yourself. And he drew the picture. This was in the first paper showing wing bud transplants. I didn't know this until much later, that Victor had written in his journal, shortly after he went to the lab, that he made this pact with the chicken embryo, that if it would reveal a secret, he would promise never to put it in a Waring blender. I followed my advisor's credo, not even knowing it was there. My second year in graduate school, I didn't go there to work with Victor. I was working with somebody else. But my then advisor came up to me and said they had persuaded Victor to teach experimental embryology. He hadn't taught it for 8 years. He was already retired. This would have been when he was 69 years old. But they needed a TA. I said, well, I can't TA. I don't know anything about that. I haven't done any of that stuff. He said, no. You can do this. And so very quickly, I had to learn how to make glass needles, make hair loops. Thank goodness my best friend in St. Louis had just had a daughter with blonde hair that I never asked the mother. I got a lock of hair to make hair loops. made thousands of glass bridges and Speymann's pipettes and all this type of stuff. And when I went to Viktor, I said, well, what are we going to do in the lab? He handed me a copy of his book. He said, we're going to start on page 1 and do everything in the book. Okay. What I didn't realize at the time is that also meant going 40 miles out of town to the Boy Scout camp and going into the ponds to collect Embystoma eggs. But that was as close as I came to a real biologist. These are from my notebook of that course. And I'm very glad I didn't notice ahead of time. There's one that you'll recognize from the first talk. We did planaria, we did chickens, but mostly we were doing fate mapping and transplants of Tarica and Embystoma. Viktor said during that time, he asked me when I was going to go to Woods Hole, and I said, well, what's Woods Hole? And he said, no, you have to go to Woods Hole. He had been 10 years at Woods Hole, course director for 5 years. And I put this in, that's Viktor on a collecting boat. I put this in, it was Mel Steinberg was the course director. Fortunately, this is real small. There's a real nerdy looking high schooler there. Looks like a high schooler, which is me, but don't look too closely. The major feature of that was that summer Lewis Wolpert was in Woods Hole. And as much admiration as I had for my faculty colleagues at WashU, I never met somebody who could use as many active verbs and adverbs in one sentence as Lewis Wolpert, patterning and French flags and positional information. And that served me well because when I got back and I had to take my qualifying exams, I could talk about things that the faculty at Washington University had not really heard about. And he had me follow up an experiment that he had done. He had extirpated neural crest and placodes, looked at trigeminal ganglia, and said, well, there's this new method that Jim Weston had just published where you could put treated thymidine, label embryos, transplant the labels, blah, blah, blah. And that's what I did. And so we did a lot of mapping. And the two major things about that is, one, I really got to reconstruct a lot of embryos with papers and do 3D drawings, which I love to do. And we also found that you could take neural crest cells from any part of the head, and if you put them back here, they would go and form a perfectly normal migratory pattern into whatever arch you put them. And so one of the things that is part of this legacy is that our forebears thought a lot about what the other people were thinking about when they designed their experiments or they interpreted their results. They didn't just think about the data. That was something that I paid a lot of attention to and continue to do. I want to mention very briefly Viktor's true intellectual mentor, and that's Ross Harrison. Viktor knew the philosophy. He rejected the notion that philosophy and science were irrevocably coupled. A philosophy was a set of beliefs that defined how you behaved and thought of yourself. Science was analytical. It was separate. It was rational. And that pragmatism defined Harrison. And I'm sure you all know the number of posters here on lateral line. He made these chimeras in 1904. And what you can see here are lateral line cells coming from, these are chimeric, pigmented, non-pigmented. There's the lateral line coming down, his famous tissue culture experiments. Harrison went every summer to Germany. And most summers he would go to Madrid and visit Cajal. And then we would come to Freiburg and spend maybe a week or so, maybe a month with Speymann's. Stay at his house, be in the lab, Viktor's thesis project was working on nerve-limb interactions. It was Harrison's suggestion to Speymann's that that would be a good project. Okay. Speymann's Harrison being the pragmatist, talked about the futility of figuring out whether something is determined and what determined it. And the reason that he said it's futile is that it's a dichotomy, and nothing in development is dichotomous. If you think it is either or, you're going to be wrong, because almost everything is somewhere along the continuum between the two. very briefly, I made maps. And when the quail, Nicole Edgar-Rand's group rediscovered the quail nuclear marker, I was just finishing up my PhD and had a job at UMass, and I set up a quail colony, did lots of mapping of neural crest cells. Nicole's group had done the low-hanging fruit, and I just kind of nitpicked around the edges. And one of the things that we found was this corneal epithelium that comes in before the mesenchymal cells come in. And so what's the big deal about this? Well, the big deal about this for me, you've seen the picture and heard about Elizabeth Hay before. She had published a paper in 1968 talking about two kinds of mesenchyme. Primary mesenchyme, like mesoderm, would form epithelia, and other mesenchyme would go back and forth, like a somite. Secondary mesenchyme couldn't do that. Never made blood vessels, never made blood cells, it never made epithelia. That was like neural crest. That's an epithelium. So what am I going to do? Those of you, we don't really have this today, but in my generation, when you were young and you were going to give a seminar, the first thing you did when you walked into the room was to see if Betty Hay was in the room. Because the one thing you knew, two things you knew is one, she knew more about what you were going to talk about than you did. She had read more literature, and B, she was going to ask you a question. And so I put the best pictures in an envelope. I did slow mail. It was the only mail available. Sent it to her at Harvard and said, well, gee, this is before I would send it off to the journal. I said, what do you think about this? It's really surprising. And she wrote back this beautiful letter. I still have it. Models come and models go. The embryo was always right. We became dear friends. I visited her often. I made maps, and I'm not going to go into details. I mapped skull parts of avian embryos. Other labs were doing the same thing. And this became much a source of consternation to comparative anatomists, and then finally markers in mice and other species came. And what's fascinating about this Devo-Evo connection, if you look at it, is that the chicken and the mouse and the axolotl and the zebrafish, in terms of skeletal elements, are virtually the same. Some of the names are wrong, but they're virtually identical otherwise. And Xenopus is evidence that extraterrestrials have actually come into the development of chordates. All right. Other maps that I made using retroviruses. And the only reason I did that was there were some controversies and more transplants were going to resolve it. And this is your mesoderm maps. What is important about this is not the maps, but the fact that the precursors of all of those different tissues were neighbors when the retroviruses went in. And within that same neighborhood, some cells, knowing nothing about their state of genetic commitment, but some of those cells didn't move, and they became cartilage. Some of them moved dorsally, and they became the skull bones. Some of them formed aggregates and moved into the periphery, became muscles. And then there were these cells, the endothelial cells. They went everywhere. And we had actually already looked at this in quail chick. There's a little mesoderm transplant of quail, and it's stained for blood vessels. These are terrorists. Angioblasts in the embryo are the only truly invasive cell that there is. They will go in whatever direction happens to suit them. And what this means as a corollary is that every blood vessel is chimeric. It has received angioblasts from multiple different parts. That tells you when the identity of the blood vessels has to not have been created and when it does is created. And the last set of experiments, we had done a lot of maps and mapped where these neural crest cells give rise to the different parts of the head. And not by design, but just of convenience, because I needed more donor tissue, we did these transplants, thinking that, well, they're going to go in and they're going to behave normally for their new environment. And much to our surprise, they didn't. These transplanted future first arch cells went into the second arch and they formed a jaw. I remember when I was looking at these and I was thinking to myself, this embryo has just answered a much more important question than I thought to ask it. These were published in '83, and the paper was not really very popular, and then suddenly Hox genes came along, and Hox gene knockouts give you a partial phenocopy of this. We looked also at muscles, and we had mapped all these muscles. There's the normal first arch muscles, second arch muscles, and we look at them in this little beast, and the muscles are changed. Around that ectopic first arch are now first arch muscles and second arch muscles going to the arch behind them. The neural crest cells, being a connective tissue lineage, just like in the body axis and just like in the limbs, in pattern formation, the connective tissue is the top of the hierarchy, and muscle and peripheral nerves and blood vessels are lower down in that hierarchy of programming. So the title that I chose originally was The Legacy of Experimental Embryology, and I was going to say, well, is this relevant to anybody today? The previous speakers have answered that question, and I realized during this meeting the question was the wrong question. Whether it's relevant or not, most of you in the room are experimental embryologists, because experimental embryology always has a comparative element. You compare one thing with another. Head, first arch with second arch, animal A with animal B. You do descriptive embryology. You folks are doing it with proteomics and reporter gene constructs and all these kinds of things. It's descriptive embryology. You're doing lineage. I never wrote a grant with the term fate mapping in it because I knew I wouldn't get funded, but I had to map all these things before I could do any experiments. You're doing exactly the same thing. You change the spatial temporal relationships, whether you do it between cell populations or you do it between transcription factor binding sites using CRISPR. It's the same basic concept. And quote of Viktor, this was '91. This is before genomics. Revolution in methods has liberated developmental biologists. Freedom allows us to concentrate on the unanswered questions posed by Boberry, Spain, and Harrison. And unknown to me when I was thinking about this, the three preceding awardees said exactly the same thing, whether they used these names or chicken embryos or their own system. You show a passion for the embryo. It was so much fun going to the talks and walking through the posters and talking to the people, because that's the essence of experimental embryology. The ones who were really great, they had a passion for the embryo. They got to know the embryo. They knew its innards, not just its outwards or just its Linnaean name. And with that, I again thank the society, and I thank my nominators. And you don't have to study the history of experimental embryology. You are its legacy. You are taking that, as Eric said, you are the people who will convey that to the next generation, whether they know it or not. That's what they're doing. Thank you.